Assalamu alaikum. I hope you're all doing fine. I'm Dr. Muhammad Ali Rabbani, and today we are going to discuss the topic of histology, cell junctions. By the end of this session, the student should be able to enumerate the various junctions that are present in cell membrane, differentiate between occluding, anchoring, and communicating junctions, elaborate the structures and functions of various junctions, and correlate the abnormalities of the junctions uh, with the features abnormal to the junction with the conditions thus produced. Okay. By now we have discussed the epithelium. So epithelium, what was the epithelium? It was aggregate of polyhedral cells that were strongly adherent to one another, forming sheaths. In definition, we discussed that in addition to that, we discussed there was on the basement membrane, plus they lined the cavities too. So I'm focusing on this part. So they are closely adherent cells, polyhedral cells that are closely adherent to one another. So they must adhere to each other by something. What is that thing? That is the cell junction. That are the specialized intercellular junctions. The intercellular junctions, they are of the following types. They are tight junctions, zonal occludence, adhering junctions, zonal adherens, desmosomes, hemidesmosomes, gap junctions, and focal adhesions. So these junctions, these adherent junctions, desmosome, hemidesmosomes, and focal adhesions. So these are adhering junctions. They help cells to adhere to one another physically and against force. These and against cell with each other and cell with the basement membrane. This one, the tight junction, this help close any passageway between two cells. This is occluding junctions, tight junctions, and this junction, the gap junction, helps two cells communicate with one another via the gap between those two. So here is a picture from Jankoira's textbook of histology. Here is the cell that is shown, to three adjacent cell rather has been shown here, and this line, this is the lateral wall, lateral domain. This wall that has been removed, this had been removed, this was apical domain, and this wall on the floor against the basement membrane, this is the basal domain. As you can well see, and it's a very common sense that the cell junctions will be between the two cells against the two lateral domains, and the cell junctions will be against the basal domain between cell and the basement membrane. So if we see at the junctions that are present from top downwards, the junctions that will be present will be tight junctions that will be present most near to the apical domain. They are present on the lateral domain, but at present at the most apical position, the tight junction, the occluding junctions, they form a ring around the cell, joining with all the cells that are near to them. So this cell, because of this ring, this is called zonula or zonula occludens. And then there is the next one, adhering junctions. Again, this is encircling the cell, so that is also called zonula adherens. Then there is desmosomes. These are points, like point weld. If you do welding of a metal on a single point, they are like that. So there are attachments on single points are present here, 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 has been shown in this picture, and here and here. These are desmosomes, and they have corresponding structure on both communicating cells. Like this is the part communicating with this part, so these are desmosomes. Then comes this one in the basal surface, the hemidesmosome, and this is the anchoring junction. This has structure roughly half of that of desmosome, because on the corresponding part there is no desmosome to communicate with, there is no cell to join with, rather the basement membrane. So there is this hemidesmosome or anchoring junction that leaves us with the one that is not technically helping the cells stay together, but that junction helps two cells to communicate with one another. So these are called gap and communicating junctions. So let's dive into them one by one. Here is a picture from textbook of histology by Michael H. Ross, and this is the picture from this is the electron microscopic picture. You will see zonal occludence, adherens, macula adherens or desmosomes, followed by gap junctions and hemidesmosomes. We will be seeing them and discussing the electron microscopic picture as we go along. Before diving into any further, for all the junctions, there will be a component that is passing the cell membrane through and through. That will be a transmembrane protein that will be attached to some part of the cytoskeleton, can be microtubules, I'm sorry, can be microfilaments or intermediate filaments. So it can be it will this transmembrane protein will be attached to skeleton through anchoring proteins. These transmembrane proteins will not be directly communicating with the cytoskeleton, will be communicating with the cytoskeletal component through some anchoring protein. So any of the junction that we will see from now on, go through these against these headings. It will be very helpful for you to memorize some things. So this is the picture again. Let us focus on the tight junctions or occluding junctions or zonula occludens. They are the most apical one on the lateral domains, and they completely encircles the cell, making junction with all the cells, neighboring cells that are surrounding the one that we are looking at. That is why that is called zonula adherens. And in in electron microscopy, the cell membranes appears very tightly adherent with no space between the cell membranes. We will see again these pictures and they seem tightly opposed. And as we saw that it has three parts. First part will be the transmembrane protein, the one that is shown here. It can be different proteins are there. It can be claudin, it can be occludin. There is a gem protein. So you don't at this level we are not going to go into the ultra structure of this protein, but you have to get an idea that this is a protein that goes through and through the cell, and it has a part that is extracellular, it has a part that is intracellular, and the extracellular domains of this transmembrane protein communicates or joins with the extracellular domain of the corresponding protein on the opposite side. That is how these two cells get attached to one another. And how this is getting attached to the cytoskeleton inside through different proteins. These proteins are anchoring proteins, the ZO proteins, zonal occludens proteins, ZO3, ZO1, ZO2, and through these proteins, this transmembrane proteins is getting attached to this microfilament actin proteins. So this is how transmembrane proteins, claudin, occludin in this case, are joined through ZO proteins to the cytoskeleton or microfilament cytoskeleton. So here are pictures from Genquera's histology. This is a picture of electron microscopy, and you can see that here the cell membranes, this is the cell. Let me zoom out too much. So this is the cell, and here there is the apical domain, and these are the cilia going upwards like here and here and here, and these are the lateral domains, two cell membranes between the cells, and then there is basal bodies of the cilia and so on. So here is the through transmission electron microscopy. Here we will see the tight junction, and as you can see, the cell membranes at this point are very tightly opposed as you compare downwards there you can see a gap between the cell membranes, and here you can almost see no gap. So this is the tight junctions. And this is a picture from scanning electron microscopy after doing cryofracture. So what is cryofracture? It is breaking the cells apart after freezing them. So if you freeze the cells and then break them apart, you can see the lines of the tight junctions along which there was the fusion. So they serve very important functions, and they ensure that cells are very tightly opposed to one another, and no, there is no paracellular transport. Thus material does not go between the cells, rather it ensures transcellular transport. So as for the tissues which have more strict control, like bladder, that does not want any tissue, that does not want any fluid to go through them, the number of occluding junctions, the number of cycles, or at least there will be more extensive, and different proteins are there to be more robust there. And other, like intestine, where water, like in colon, where water absorption is required and we want fluid to go through them, then at that point the occluding junctions are not that much tight, not that much extensive, and they are not 100% waterproof. Not only regard transcellular transport, as you have discussed in the lectures of cell physiology and cell anatomy, the cell membrane is a very fluid structure with phospholipids floating around, and in between them proteins are floating around. So fluid mosaic model, as you call it. So due to this tight junction, this membrane is divided into apical domain on the top and basal and lateral domains, basolateral domains. And the proteins and lipids, they usually do not cross the barrier of this occluding junction. So the cell membrane can have very different features on the apical domain as compared to its basolateral domain. Also, some structures after cell make connections, or this junction join, and different signaling molecules are connected to the actin filament through this junction too. Applications: some diseases like food poisoning caused by Clostridium perfringens. This is caused by enterotoxin produced by this organism. So this enterotoxin that is produced by Clostridium perfringens may bind to the claudin molecules of intestinal cells, and that alters its structure, leading to some loss of function, and those tight junctions cannot perform their function as well. So the fluid is lost through paracellular pathway into the intestinal tract, that leads to the symptoms of diarrhea. Similar situation arises in the Helicobacter pylori infections, when it targets the anchoring proteins of ZO1 of the tight junctions and disrupt the signaling, and it alters the features of epithelium, leading to loss of fluids and ultimately takes the disease to ulcer formation and so on. Similarly, there is a lot of diseases much more than that. Similarly, loss of occluding junctions can compromise blood-brain barrier if some particular proteins are damaged, leading to neurological disorders too. So this was regarding zonula occludens. Now fascia occludens. This is somewhat extension of zonula occludens. These are ultrastructurally similar to the zonula occludens or tight junctions, but they do not have this zonula part, meaning they do not make a continuous circle around the cell, rather there are discontinuous interrupted strips around the cells. So these are somewhat found between the endothelial cells. In the blood vessel cell, the endothelial cells are like this. They are present very closely adherent to one another. So if like this, there are many cells that are present here. If the junctions are present, then there is a gap, then junctions are present, then there is a gap, then junctions are present, then there is a gap. So this allows the fenestrated capillaries to work where there are gaps between the cells and allow two molecules to pass, and the concentration or the size of the gaps determine what structures will pass through. but that does not hold true for the brain, where the connection between the cells is very tight, allowing very little material to pass through it. So this was about zonula occludens. Next is zonula adherens or adhering junction. This is the one that encircles the cell immediately below the zonula occludens or tight junction. It provides tensile strength and support junction above, because zonula occludens, although it is preventing the material to go through them, but it is not that strong that it can be broken easily. So to give it support, there is a supplementary junction just below it. It firmly anchors the cell to its neighbor through actin cytoskeleton, or as you know, that is the microfilaments. Then this junction is mediated by following proteins. As you saw, that there will be a transmembrane component, there will be an anchoring component, and then will be a cytoskeletal component. The transmembrane component in this case are called cadherins. These are proteins that bind to each other in presence of calcium. They have extracellular domain and extracellular domains of neighboring cells. Extracellular domains of cadherins of neighboring cells join with one another through calcium, in presence of calcium rather. And then the intracellular domains are connected to catenins, and that are connected to actin binding proteins. There are a number of proteins, prominent among them are vinculin and actinins, and they join catenins to actin filament, that is the cytoskeleton, forming the terminal web just beneath the cell membrane. So here is a picture from Michael H. Ross textbook of histology, Ross and Paulina, and here is the cadherin molecule shown here, and this is the extracellular domain of the cadherin molecule, and the neighboring cells, they join to one another in the presence of calcium, and the intracellular domain is attached to catenin through vinculin, and actinin, it's joined to actin terminal web. So this is how the adhering junction is formed. Now, bringing back you to the same picture, and here you will see adhering junction right here, and as you can see, compare it with the tight junctions, there is a little bit of gap that is shown between the two cell membranes. And as you can see, just below the cell membranes, you can see this dense area. It's made up of those anchoring proteins that are making up this dense area. Then there is fascia adherence. Just as fascia occludens was to zonula occludens, fascia adherence is to zonula adherence. It has the structure similar to zonula adherens, but it is present as small strips rather than a complete zonule surrounding the cell. And good example of it is presence of uh, fascia adherens in intercalated discs in the cardiomyocytes. Sometimes these are lost, and that allow the cells to break away from one another, and that is very true in the case of tumors. In the case of tumors, the key adherents are lost and the zonal adherents are lost, that leads to cells shifting away from its epithelium and migrating to develop a malignancy. So now we have done tight junctions and adhering junctions. The next one is desmosome, macular adherens. These are like these spot welts. How are they made? Disc-like shape. They have corresponding structures on the opposite cells that are joined in the midline, in the cell membranes, in the lateral domains, and they are the ones that are connected to the intermediate filaments. Up till now, the occluding and adhering junctions were connected to the actin or microfilaments. These are connected to the intermediate filaments. Intermediate filament is making up the web of material, or the web of cytoskeleton inside the whole of the cell, including the cytoplasm and the cell membrane. And near the where they are connected to the junctions, they are joined together as a bundle, and these bundles are called toner filaments. So these are the points where actually stress is delivered to the desmosomes. They contain, just like the same as we did, will have a transmembrane component, they will have an anchoring component, and they will have a cytoskeleton component. The transmembrane component are most common proteins are desmoglein and desmocollins. These are in fact types of cadherin. These are types of cadherins, and these are transmembrane clustered transmembrane proteins. These are then attached to placoglobins. These are catenin-like proteins. They are attached to the intracytoplasmic intracellular aspect of desmoglein, desmoglobins, these cadherins. Placoglobins in turn is attached to desmoplakins, and desmoplakins is in turn attached to the intermediate filaments. To make things simple, these, I know this is a mouthful of material, difficult to say and memorize, but just you can collect these two things, desmoplakins and placoglobins, as desmosomal attachment plaques. And if you want to memorize simpler things, just memorize cadherins on the transmembrane proteins. Then there is a plaque, and that plaque is attached to the cytoskeleton, that is intermediate filament. That is the bare minimum that you should ideally know. So let's just see that on a picture. This is a picture again from Ross and Paulina textbook of histology. The one that is shown in the yellow is a transmembrane protein, that is the cadherin component, that it can be desmoglein and desmocollin. In the midline they overlap with one another, fusing and making the junction, and in this intracellular component that is attached to these anchoring proteins, together they are making the desmosomal attachment plaques. So these are the desmosomal attachment plaques made up of globins and desmoplakins, and that plaque is in turn attached to the intermediate filaments. And here, as you can see, there, due to the extensive nature of the extracellular component of this desmoglein and desmocollins, the cell membrane is a little far apart, and due to the overlapping nature, you can see this overlapping nature is visible at this line. As you can see, this line, this is the whole desmosome, and between two cell membranes there is additional line that is visible, and this is due to this overlapping nature of this cadherin proteins, that are desmocollin and desmogleins. And here, this part, this is the intracellular attachment plaques that are prominent here, and according up to extending up to this, and this, all these fibers, these are basically the tone of fibrils that are transmitting the tensile strength of the cytoskeleton to the desmosomes. So here is another picture from Gencoira's histology. Almost all of the same things are shown here. You can compare it with these tight junction, adherent junction, and these desmosomes. The center line is a little visible right here too, and all of this have been discussed. And various blistering diseases. This particular picture is from Pemphigus vulgaris, and various diseases are caused due to autoimmune reactions against desmogleins that leads to the cell detaching from one another and cells becoming separate from one another. So this usually involves epidermis and stratified squamous epithelia. So this was regarding desmosomes. The next one on our list is hemidesmosome. This is the anchoring junctions, and these attach the basal aspect of cell membrane to the basal lamina or the basement membrane, and it is very much like the desmosome ultrastructurally, but there are a few differences. In desmosomes, the intra though in desmosomes the transmembrane component was made by cadherins, those were desmoglein and desmocollins. Here, the transmembrane component is through integrin. So they are integrin proteins rather than cadherins, and through different proteins, they are ultimately getting attached to intermediate filaments. Extracellularly, let me show you in the next picture. So the transmembrane component, this one, this is the integrins. This is different from desmosomes where there was cadherins, desmoglein, desmocollins. Intracellularly, there is again a anchoring plaque, intracellular plaque made up of different proteins. Desmoplakin and plectin is important here. In the desmosome, there were desmembrane and desmoplacoglobins and desmoplakins. Here, there is plectin and desmoplakins. So that is, if you don't memorize the name, don't worry, at least memorize that there is an anchoring plaque that is between the integrins and the intermediate filaments. Extracellularly, it has to attach to the basement membrane. Basement membrane or basal lamina, it's majorly made up of collagen type 4. It is majorly made up of collagen type 4 bundles. So this is attached to laminins, the one that are shown here. These are attached to laminins, and laminins then through various proteoglycans, perlecan, nidocaine, they gets attached to the collagen type 4s. So that is how hemidesmosomes extracellularly connect to the basement membrane through laminins, and intracellularly through anchoring protein they get attached to the intermediate filaments. Focal adhesions or focal contacts, they are also basal junctions joining the cell to the basement membrane. They do resemble with the hemidesmosome, but they differ a little as they are smaller in size, more in number, and they attach to the microfilament part of the cytoskeleton rather than intermediate fibers part of the cytoskeleton. So they have intermediate anchoring molecules. Majorly pexilin is the anchoring molecule between the cell, the junction, and the transmembrane component and the actin component. So they have integrins as cellular transmembrane part, pexilin as anchoring part, joining to the actin as cytoskeleton part. These are anchoring and these are transmembrane. So here is the picture again from Ross and Paulina showing this integrins, and joining inside there is a anchoring proteins. A large number of cells are given. You memorize one of few of them to at least survive the exam: taline, pexilin, vinculin, alpha-actinin. So these are all anchoring ones, and then this is attached to the actin filaments. So we have done all these. The last but not the least, there is a communicating junctions that are the gap junctions, nexus. These are the ones that mediate the intercellular communications, and rather than getting the cell together, they make cells talk to one another. And there is the only known structure that actually allows direct passage of signaling molecule between cell to cell without having to go out of the cell and then coming back. And due to their size, they allow for very small molecules, roughly 1.5 to 2 nanometer in size, and they're very important in the cells where the cells have to act in synchronously, like smooth muscles. Through gap junctions, ions are transported and they contract in synchronously. And similarly in cardiac muscle, through gap junctions, the ions move around and the action potential is moved to the next cell, and thus heartbeat as a whole. So in most of the cells they are present where cells have to work in unison. They are present there. You have to memorize a few words. There is a protein. They are made up of protein called connexin. This single protein, this single protein, this is called connexin. This single protein, this is called connexin. 6 connexins make up 1, 2, 3, 4, 5, 6. 6 connexins arrange themselves in a tubular manner to make a tube that is called connexon. That is a variable diameter depending upon the exact nature of the gap junction. This connexon has a corresponding tube on the opposite cell, and they align with one another to form a whole channel between two cells. So there is a pair of connexon joining the two cells, and then there are hundreds of connexon aggregated together, and this whole aggregation of multiple connexon pairs, this whole is known as gap junctions or nexus. This circular patch is called gap junction. So this is a picture from Ginkoera, and here is again these are shown, and this is a picture from cryofracture, breaking the cell apart and showing you this is the gap junctions. Okay, so that's it, and they open and close. Some gap junctions open and close too with response to different stimuli, like in the presence of high calcium level they close, in the presence of low pH or high hydrogen level or proton level they close, and there are various triggers and you will discuss them in physiology in detail. So that's it. Before closing the lecture, let us try to check if you have, let's pause the video and try to answer these questions. Your instructor reminds you of the epithelial cells have membranous channel that permits ions and small signaling molecule to pass between the adjacent cell. Which of the following proteins from these form these intercellular gap junctions? So we're talking about gap junctions already mentioned here. So what proteins are making up? So these are made up of connexins. Adherens are the transmembrane proteins, perforins are the aqua water channels, and there are other two, but connexins are the ones that are present in gap junctions. MCQ number 2. A 25-year-old woman spends an afternoon raking leaves. Later that evening, she discovers fluid-filled blisters on the palm of her hand. Leakage of fluid from dermal capillaries at the site of minor injuries of hand of this patient are regulated by changing in which of the following intercellular junctions? So, leakage of fluid from dermal capillaries. Leakage of fluid. Which junction was preventing the leakage of fluid? So the leakage of fluid was regulated by occluding junctions. So zonula occludens was responsible, so that was damaged somewhat due to that fluid was filled in. Which of the following membrane junctions anchors the epithelial cell to the extracellular matrix molecule in the underlying basal lamina? So the answer is hemidesmosomes. Hemidesmosome or focal adhesions are the ones that join the cell basal aspect to the basement membrane. So that is it. Thank you so much for listening. I hope that was helpful for you. If you have any questions, suggestions, or feedback for this lecture, please do mention in the comments below, or you can contact us with any of our social media handles. The lecture recordings and presentations are present on this website, medicosacademics.com, and you can follow us on Facebook and YouTube. Till next lecture, see you next time. Allah Hafiz.